Hello, my name is Aman Patel, and I will be presenting our work on hyperbolic geometry-based deep learning methods to produce population trees from genotype data. So a recent trend in biomedical sciences is that there's been a lot of work on predicting disease risk and other important traits from an individual's genomic data. The problem is that these models often generalize poorly across populations. If a model is accurate for one group, there's no guarantee it'll be accurate for another group as well. It's therefore important to know an individual's genetic ancestry to make the most effective decisions possible in terms of healthcare. One very important question is whether a study from one group will generalize to another group or not. We therefore need a method to represent ancestries and how related they are to each other. Keep in mind, ancestry is intrinsically hierarchical. At various levels, we can, for example, classify someone as European, Italian, or Sicilian. Therefore, trees represent the ideal data structures for this type of application. And in practice, these are often produced directly from the genotype data using some measure of relatedness. However, we run into a big problem here. As genomic data sets get larger and larger, trees will be more and more expensive to create. What's more, they're non-parametric, meaning that adding new data points requires a recreation of the entire tree. And furthermore, existing methods also don't really integrate well with current workflows, particularly in machine learning. We aim to solve these problems by developing deep learning methods to create continuous embeddings of population trees. As shown in this schematic on the top left, new points can easily be added to our embedding spaces without retraining the entire model, thus vastly reducing computational costs. A simple and inexpensive heuristic can then be used to produce an actual tree if that is desired. And we specifically use hyperbolic space for our embeddings because it's long been known as the ideal space to represent hierarchically structured data. In 2020, a method called HypeHC was released, which defined a loss function to embed data in hyperbolic space such that the structure of the embeddings reflects that of a hierarchical clustering tree. In this project, we combined the HypeHC loss with two different architectures. First, a multi-layered perceptron, or a fully connected model, in which the loss function is the HypeHC loss. And second, a variational autoencoder. Here, we add the HypeHC loss to the standard VAE objective, and we also make some other modifications to adapt the model to hyperbolic space. In all experiments, our models were trained on a data set of over 5,900 genotypes that were derived from 136 different ethnic groups across 6 continents. Our first objective was to see how well these models generalize, and for that, we first generated trees using only the test set, so on completely unseen data. And in both cases, we separate the continental groups from each other pretty effectively, and the way the populations are split reflects known human evolutionary history. As expected, for example, the African population is the most isolated, and the order of the others is very reflective of how we spread out as humans after migrating out of Africa. We next wanted to check if new data points can be effectively added to an existing tree, since this was one of the most important selling points of the model. When given a tree produced by the training set, or produced from the training set, both models were still able to add points from the test set in a semantically meaningful manner. That is, extra data points fell well within the clusters that we would have expected them to fall into. This was even true for populations like the Oceanian population, which didn't have very many training examples at all. And these results demonstrate, once again, that the models generalize well and that new data points can be added effectively. Finally, we performed a comparison between the quality of the trees produced by our methods and those produced by more conventional hierarchical clustering methods. To do this, we used Dasgupta's cost, a traditional metric for hierarchical clustering quality. Note that the benchmark methods here were fitted directly on the evaluation data, while for our methods, that data was unseen. And despite this, our methods performed comparably to the benchmarks, and even in some instances, outperformed them. This indicates that even on unseen data, our methods are producing accurate trees and falling well within the ballpark when compared to the established methods. In conclusion, we have designed methods for population tree creation that integrate with larger machine learning models and workflows, accurately incorporate unseen data, and perform comparably to established non-parametric approaches. As biobanks and other genomic repositories grow in size in the future, our model's flexibility to iterate on existing trees and add new data points will be essential. Our results have also uncovered several potential further directions that may be very promising. For example, we believe that by simply adding more training data, we can get even better clustering between continental populations and more effective clustering with subcontinental populations. We have not also explored all the various capabilities of the VAE model, and doing so could potentially be very powerful. And finally, the HypeHC loss actually allows for a lot of flexibility in terms of injecting additional data in to the model that is not found in the input genotype data. Fully determining all the possibilities here could also be an intriguing area of future exploration. That's it for the presentation, and thank you very much for listening.